the mouse? All right. Thanks, Diane. No problem. So what you're looking at here is you get massive tissue iron accumulation, and it can happen in very young mice. And so then the question was, how does intestinal VHL regulate aspects of systemic iron homeostasis? And so to give you a little bit of background on what VHL does is that it is known to regulate hypoxia signaling. And I put up these three individuals. They were awarded the Nobel Prize in Medicine in 2019. Those that did hypoxia work, we were ecstatic because it highlighted our field. And so Greg was the investigator who identified a transcription factor that was regulated by oxygen tension. And so the way he did this probably was known to many of the audience is that he took at that time what was the only known oxygen regulated gene, which was a gene called erythropoietin. And so this gene is critical for the synthesis of red blood cells. So what he did was he took the promoter of this gene and basically threw on gallons and gallons of lysate to see what could be pulled down. And he identified a transcription factor that he aptly named hypoxia-inducible factor. And then this discovery was made in the late 80s, early 90s. And another point is that this discovery wasn't well appreciated at the time. He actually published this in JBC and MCB. And so in the next 20 years after that discovery, Bill Kalin and Peter Ratcliffe discovered how this transcription factor was regulated by oxygen tension. And so what I'm showing you here is really about 30 years of work wrapped into one slide. And so what they discovered was that this transcription factor that Greg discovered, hypoxia-inducible factor, had two proline residues that can become hydroxylated. And once it's hydroxylated, this factor that I was interested in, von Hippel-Lindau tumor suppressor protein, or VHL, could bind to HIFs and lead to its degradation. And the reason this was oxygen regulated was that they discovered a group of enzymes. Now we know that there's 3 of them called Ph.Ds 1, 2, and 3, also known as EGLN 1, 2, and 3. Could hydroxylate HIFs. And the way that they hydroxylated HIFs is that they use molecular oxygen for their hydroxylation reaction. So once HIFs were hydroxylated, VHL would bind, leading to its rapid degradation. However, in scenarios where oxygen was limiting or VHL was mutated, HIFs could become stabilized and then bind to a heterodimer partner called HIF1 beta or ARNT and regulate the transcription of a whole host of genes that were critical to regulate adaptation to hypoxia. And one of the first meetings that I attended when I first started my lab was this Keystone Hypoxia meeting in like 2010 or 2011. And these were the shirts that they were handing out. Basically, almost every pathway that we can think of may be regulated by these transcription factors. And so really, if you boil this down into what are the group of genes that these transcription factors regulate, I think it comes down to 3 genes. One is genes that are critical for cell metabolism. So once a cell becomes hypoxic, it's very difficult for it to undergo oxidative phosphorylation efficiently. And so it mainly uses glycolysis as its major means to produce ATP. And most glycolytic genes are HIF target genes. The second group of genes is oxygen delivery. So as you can imagine, when a cell becomes hypoxic, one of the adaptive processes, how do you deliver oxygen more efficiently so it can become more normoxic? And so there's a number of genes that fall into this category. Erythropoietin is one, which regulates systemic red blood cell levels. But another group of genes that fall into this category are angiogenic factors that can now build blood vessels to deliver oxygen to hypoxic foci. And so almost any angiogenic factor that's been identified as a HIF target gene, and one of the well-characterized ones are vascular endothelial growth factor alpha. And then the next group of genes that fall into this category are genes that are important for cell proliferation and cell survival. And so as you can see, based on these group of genes, why this transcription factor was studied so heavily in cancer, because all of these genes are critical for allowing cells to grow, I should say cancer cells to grow in harsh environments, especially in nutrient limiting, hypoxic environments. HIFs are absolutely essential in those scenarios for the cells to grow and maintain viability. It becomes even a little bit more complicated after Greg discovered HIF-1 alpha. Several years later, three groups had converged and identified another gene that had very high homology to hypoxia-inducible factor 1 alpha. And then they aptly named that gene HIF-2 alpha. What you need to know is that these fairly homologous, this is what we call the oxygen-dependent degradation domain. So these are that contain those two proline residues that get hydroxylated. They're highly homologous between HIF-1 and HIF-2. So they're regulated in the same fashion, meaning VHL-mediated or proline hydroxylation, VHL-mediated degradation take place for both of them. They both heterodimerize to this partner called ARNT or HIF-1 beta, and they actually bind to the same response elements and the promoters of genes. Through our work and through several other laboratories, what we've discovered is that actually they regulate a different subset of genes. And so now if you look into a given cell, in a given cell, about 30% of the genes that HIFs regulate are overlapping, meaning HIF-1 and HIF-2 can regulate the same subset of genes. However, the majority of the genes are selectively regulated either by HIF-1 and HIF-2. And the short conclusion of everything that's been done with the selectivity is we don't know exactly how HIF-1 or HIF-2 regulate their subset of genes. And that's an ongoing work in my lab that I won't really discuss today, other than to tell you that everything that I'm going to talk about is exclusively regulated by HIF-2. None of it can be overlapped with HIF-1. And we're still trying to work out some of those more mechanistic details. And I think I should mention, we can keep this informal. So if you have questions throughout, feel free to blurt them out. I have plenty of room to address questions in this talk. And so what we've done, I shouldn't say just what we've done, what several labs have done in this space of understanding how HIF and specifically HIF-2 regulate aspects of iron homeostasis, we've published in the past 10 years. And so I've really summarized it into this slide of what we've done. And so I've mentioned that there are apical transport systems and basolateral transport systems that are required to move iron into the enterocyte and move iron out. And every one of those genes we've demonstrated to be a HIF-2 target gene. And so if you knock out HIF-2 in those scenarios, those genes are not aptly upregulated. And so in the long run, if you knock out intestinal HIF-2, they will become spontaneously anemic as a function of time. And those genes that are needed to upregulate the movement of iron into circulation are not regulated as you would see in a wild-type mice. We've now recently also have integrated HIF-2 regulation into pathways of moving iron out of ferritin known as ferrintophagy. And so we've identified those genes that are regulated or needed for lysosomal degradation of ferritin to make iron available at the cellular level are also under the regulation of HIFs. And so at this point, I kind of tell my lab and anyone that's listening to me that cancers don't discover new biology. We think that they co-opt it from normal pathways or developmental pathways or adaptive pathways under stress. And so this is when we started beginning, look, if HIF is absolutely essential to move iron into enterocytes and to maintain systemic iron homeostasis, could any of that regulation be hijacked by cancers? Because we know that cancers are highly hypoxic. And so here's what I'm showing you really is a cartoon version of how we view cancers. So we view cancers as a pseudo organ that's not only made up of cancer cells, but made up of a whole number of ancillary cells that really predict how fast that tumor is going to grow and how it's going to respond to therapy. And this environment is highly hypoxic. And so then the question was, if it's hypoxic, most likely HIFs are upregulated, specifically HIF2. Can any of these aspects of what we demonstrated with respects to iron homeostasis be co-opted by cancers to either increase growth or affect treatment to response? And so I put this in there. My hypothesis is that we think that iron plays an essential role in maintaining cancer growth. And just to show you that we're a GI lab and we focus on GI specific cancers. And one of the cancers that we focus on that we know HIF2 is absolutely essential for is colorectal cancer. And one of the models that we use, in addition to other models that I'll talk about a little bit later in the presentation, is this model of carcinogen-induced injury colon cancer model. And the way this model works is fairly simple. We give them a low dose of a carcinogen called oxymethane or AOM, and then we follow it up with bouts of injury by a compound called dextrane sulfate sodium. And so this is a colytic agent that causes massive injury in the distal intestine. And so if you cycle in this injury agent several times after giving this carcinogen, the mice will induce colon tumors. And so if you did this in the background of knocking out either HIF1 or HIF2 in intestinal epithelial cells, what you'll see is that knocking out HIF1 has no role in colon tumor genesis in this model. However, when we knock out HIF2, we can impact tumor number, tumor growth, tumor proliferation. I'm just showing you tumor number. We published a number of papers in this space. So this cancer is dependent on HIF2. Moreover, some of the work we're doing now, both in the space of iron overload and also in the space of colorectal cancer, is that there are very good FDA-approved inhibitors of HIF2 that are now in phase 3 clinical trials for other cancers, and we're testing their efficacy in our colorectal cancer models as well. So the question was, do these tumors acquire more iron? And is that iron important for their growth? And so really probably not new to this audience, you can measure cellular iron in tissues using ICP-MS. We now have a standard protocol that we actually use quite a bit, where we take normal and tumor tissues and we run them through what we call our metallomics platform, where we measure a handful of metals that are biologically relevant.
So Yatrik?
Yes.
You said we can ask questions. What
for sure. How are you?
Okay, good. What happens to the homozygous knockout mice? Because you're looking at the heterozygous.
You're talking about when you knock out HIF2?
Yes.
Well, actually, this model that I'm showing you is a biallelic knockout of HIF2 just in intestinal epithelial cells. Now, if you make a whole body HIF2 knockout, they're embryo lethal very early. So that's a very good point that I forgot to mention. This is an intestine epithelial specific knockout HIF2 that decreases tumors. HIF2 whole body knockout is not viable with life in most backgrounds. Now, you can get some mice that are born in certain backgrounds And no one, I would say that is very, I think survival is decreased by 95%. So you lose most mice if you make a complete HIF2 knockout. But there's some mice that can bypass the need for it. And we're starting to figure out exactly what those mice, how do they bypass it? But in most cases, knocking out HIF2 is not viable for life. And I should say the same thing for HIF1. If you knock out a whole HIF1, they're not viable as well. And the same can be said if you overexpress these transcription factors. So if you knock out VHL embryonically, that's not viable with life either.
Right. So this is a deletion of both alleles. Delta
This
E.
is a deletion of both alleles.
Thank you.
Or, I don't know if we have any data, if we just lowered HIF2 with one allele, what its role in colorectal cancer would be.
Thank you.
But thanks for that clarification. I forgot to mention it. So the bottom line is that there's a lot of metals that are different in cancers versus normal tissue. We can assess this by both ICP-MS, and it's actually what we found when we've gone through this pipeline. We've actually measured a number of cancers, and I could say that our iron and copper phenotype, I would say, is fairly well conserved between almost every cancer that we've measured. The only cancer that I can tell you right now that doesn't have an increase in tissue iron accumulation is gastric cancer. And I won't go into the details of why we think that, but most cancers accumulate high levels of iron. We do an orthogonal approach to make sure that this is indeed correct. And we use Prussian blue for that. And the reason is iron, if you get a hemorrhage in a tumor, which is not uncommon, it will throw your iron numbers off in ICP-MS. And so it's nice to get some histological data to make sure that we're not looking only exclusively at hemorrhage tumors. So you can see, even doing Prussian blue, we can see the area of iron staining is much higher in patients that have colorectal cancer compared to normal. And we can demonstrate, at least in the mouse model, this is dependent on HIF2. So if you take normal and tumor tissues from AOM, DSS, or even our sporadic models, you can see an increase in iron accumulation in the tumor compared to normal. And if we knocked out HIF2, we can bring it back down to baseline, demonstrating that HIF2 is absolutely essential in this iron accumulation. And so then the next question is this iron critical for the growth of these cancers? And again, I'm going to go over this data very fast. You can knock out the apical transporter and stop iron delivery into tumors. And the way we made these mice is that we bypassed the small intestine, because that's where a lot of the iron absorption happens. So we use a promoter that's exclusively activated only in the colon. And so we're knocking out DMT only in colon or colon tumors, and we can see a decrease in tumor numbers. And here we can actually see a decrease by just knocking out one copy. We can then, also show if you throw in high amounts of iron, you can potentiate tumor genesis. And again, aspects of that are blocked by knocking out DMT. What about the opposite? You can knock out ferroportin, which is your iron exporter. So if you knock it out in colon epithelial cells, those cells will accumulate high levels of iron, as we can show in this graph here. So again, what you're looking at is normal versus tumor. We see iron levels go up, which is what we've shown previously. However, if you knock out ferroportin, even in our normal tissue, you can see high levels of iron. And in tumors, it goes up even more. And if you now looked at tumor number, tumor size, or growth, if you accumulate iron in epithelial cells and in tumor epithelium, you can potentiate aspects of colon tumor genesis. I should mention that the tumor numbers, or I should not, the tumor size that we see in these mice when you knock out ferroportin, we've not seen it in any other mouse model that we've investigated. These tumors are massive compared to mice. So it robustly potentiates aspects of tumor genesis if you can accumulate iron in these tumors. So we've addressed if you manipulate the iron levels.
Hey, Yatrik,
Yes.
I got a question for you.
Shoot.
So-
How are you, Iqbal?
I'm doing well. So were you surprised that DMT1 was actually expressed in the colon under those conditions?
It's a good question. No, we weren't surprised, because when we did, so the first things that we started doing at that point, when we're trying to figure out exactly what HIF2 was doing in our tumors, DMT was the most differential regulated gene when you looked at tumors from a wild type or tumors from a HIF2 knockout. So we knew that there was some expression. And then one of the other pieces of data is that if you looked in patients, actually normal versus tumors from colorectal cancer patients, DMT is one of the most, I shouldn't say with respects to fold change, but if you looked at p-value between different patients, DMT is one of the most significant genes that are upregulated in colorectal cancers from patients too. And so we had an idea that it was getting upregulated in, at least in the tumors at that point.
So the upregulation is a consequence of HIF regulating and turning on DMT1 in the colon? Is that what
Exactly. I think that's what's happening. And so, again, this comes back to my comment that we don't think cancers discover new biology. They just co-opt either adaptive or stress pathways or developmental pathways. And so normally hypoxia-inducible factor would increase HIF2 in the small intestine to regulate iron absorption. And cancers become highly hypoxic that upregulate HIF2 to regulate at least iron delivery in the tumors.
But wouldn't you expect that colon should be almost hypoxic? I mean,
Yeah, it's another great question. And so when you look at tissue oxygenation, it's quite dynamic between different tissues. So what I mean by that is if you take a lung versus you take a colon, they're going to be dramatically different with respects to their oxygen levels, right? The partial pressure of oxygen is quite different in between those tissues. But at the same time, their HIF regulation is quite different. And so if you take a lung, you're not going to find much HIF there, which you would predict. But if you take a colon, it's very difficult to find HIFs there as well, right? And so the way we hand wave or speculate that is that I think that every tissue has a different threshold for HIF activation. And that could be predicated by the Ph.D. enzymes or other mechanisms that impinge on the degradation of HIF. And so not all hypoxic tissues will have very high levels of HIF. Cancers are even more hypoxic than the normal colon. And once you get into those severe hypoxia, then HIFs get maximally activated. That's my explanation. I think we need to do a little bit more with respect to experimentation, but certainly if you look at HIFs in different tissues, I think you would see that even though some tissues we consider to be hypoxic, so for example, chondrocytes in the growth plate are very hypoxic. That's an avascular tissue. Colon is very hypoxic, but the level of HIFs are not dramatically different compared to tissues like the lung, which are more oxygenated.
so have you ever thought about therapeutically turning on DMTs and ferroportin such that you can now able to increase iron absorption and iron deficiency?
There's a paper that I won't discuss today. It was actually discovered independently by my group and Fudi Wang's group in China that using roxadustat, also known as FG4592, can improve aspects of anemia in mouse models. So basically a TMPRSS6 mouse that's highly anemic, we can almost completely cure the anemia by giving activators of HIFs. And it reminds me, I forgot to mention it, we can do the opposite in patients that are in, at least I should say, not patients, in mouse models of iron overload, we can give HIF2 inhibitors and decrease tissue iron accumulation. And so both of those works we're starting to try to figure out if we can do clinical trials in patients as well. And so the next question is, what's the iron doing? Why is it so important? And again, this is not to say it's doing one thing. Obviously, I mentioned that it's involved in what, like 40 to 50% of all enzymatic reactions. So iron has a central role in cellular metabolism and in cellular homeostasis. And so these are a few things that we think that it does in a cell. It's absolutely essential for mitochondrial respiration. When you look at those proteins that are essential for moving electrons in the electron transport chain, many of them are heme or iron sulfur containing proteins. So we think that it's regulating many aspects of cancer metabolism for cancer cell growth. And so again, I'm going to show you very quickly a few pieces of data of what we've done. And so what you can do is you can manipulate iron levels and ask the question of what are the metabolic pathways that are regulated in a tumor. And we do this using a platform that we call snapshot metabolomics. So basically it's LC-MS metabolomics, where we're getting a snapshot of what's happening, and we can measure somewhere about 250 to 300 metabolites in a cell. That was to give us an idea of different metabolic pathways, such as glucose metabolism, urea cycle. And so when we do this, the overwhelming pathways, or I would say the majority of the metabolites that come up are in nucleotide synthesis pathways, which again are not completely novel. We know that many enzymes that are involved in nucleotide synthesis require iron. But what was interesting is that we can take this model and we can understand how important is iron in regulating the growth. And so what we do here is we use primary tumor cells. We can do this in cell lines as well, but I get called out every now and then of being very dependent on cell lines. And so what we've done is we've taken patient-derived, what we call enteroids. So basically these are primary cells from patients. We can take the tumors, we can grow them, and now we can manipulate iron and ask what happens. And the other advantage to these models is that we know in our bank, and this is actually a plug for our bank, if you're ever interested in doing enteroid work, we have a large amount of colorectal cancer and even normal enteroids frozen down. And we've done the mutational landscape of these models. And so if you look at the mutational burden, it's quite different between different lines, right? Some lines have an APC KRAS. We have a line that we call, it's a sessile serrated polyp, which is actually doesn't have an APC KRAS mutation. You take these lines, you can remove the iron using DFO and doses of DFO that don't completely kill these enteroid models. And we can now replenish the nucleotides by just throwing on a bunch of nucleosides in these culture media. And we can partially rescue some aspects of growth. So again, I'm not telling you that iron through nucleotide metabolism is doing everything, but it partially explains exactly where and how iron is being used in cancer cells. And to really briefly summarize this part is that we know iron accumulate in tumors. Specifically, I just showed you colorectal cancer. The one thing that I didn't show you that I'll get into the next part is that somehow these tumors can bypass the toxicity of iron. And we think that iron regulates aspects of metabolic function and cancer metabolism in these cells that we're still further exploring. So the question was, it's simple. You deplete iron, you can decrease growth. But the issue with that paradigm was that you decrease growth, but as soon as the cells can get a hold of iron, the growth comes back to normal. And so in our models, we can only decrease or have them, we halt or decrease proliferation, but we don't kill cells when we decrease iron levels. And the question was, can we utilize the dependency of iron in these cancers to come up with ways to kill cancer cells? And so what we did here was kind of an in vitro screen where we made enteroids from what we call our tumor model, which is just APC knocked out. This is the initiating mutation in colorectal cancer. Or in vitro, we knocked out APC and we manipulated HIF2. Now we can activate HIF2. And so these enteroids actually have HIF2 overexpressed, what we feel like what would happen in vivo in cancers. And so then what we did was we cultured them and we threw on a bunch of different inhibitors, chemotherapeutics, and things that we were interested in doing a small drug screen using enteroids. And one of the things that we found, and so if you look at just from the APC knockout, you have what we call our sensitivity plot. Some drugs work really well, some drugs don't work well at all. And one interesting thing is if you did this in mice or enteroids where you had HIF2 overexpressed, some of the drugs that didn't work that well are actually highly sensitive when you overexpress HIF2. And one of the group of drugs that we found are these drugs that are important in the initiation of ferroptosis. And so what is ferroptosis? As the name suggests, it's an iron-dependent cell death program that's independent of apoptosis. I'm not going to get into kind of the weeds of what I think about iron in this pathway, but other than there is some dependency of iron, but really the hallmark of this cell death pathway is the accumulation of lipid ROS that leads to lipid peroxidation mediated cell death. And now we have a number of reagents that can manipulate this pathway. So you can artificially activate this pathway using drugs that can either inhibit this glutathione peroxidase that's important for removing lipid peroxides, or you can activate ferroptosis by inhibiting this transporter that brings in cysteine into a cell. And all of the goal of this pathway is to limit lipid peroxides. And so the drugs that we had identified in our screen that were exclusively sensitive when you overexpressed HIF2 were Avastin, RSL3, and another drug right here, sorafenib. And so it gave us a clue wondering if HIF2 was important in increasing the sensitivity to ferroptosis. I'm going to just show you one piece of data that suggests indeed that if you can activate HIF signaling, and specifically HIF2, they're very sensitive to ferroptosis. And so what you're looking at is colony forming assays. And so each dot is a cancer cell growing in a colony, and we stain it with crystal violet so they turn purple. And so the more purple, the more cells that are around. So you can see you take control cells, they grow just fine. We use our Ph.D. inhibitor, so we're artificially activating HIF signaling, which we covered a little bit. And so this is roxadustat or FG4592. The cells grow just fine in them. If you throw in Avastin, we're using a dose where cells can still grow, but we have some effects on colony forming. But when you combine Avastin by activating HIFs, we can completely decrease growth. And I'm not going to get into the details here, but this is specifically mediated by HIF2 and not HIF1. And you can do the same thing by just overexpressing HIF2. And so then our thought was, if we can figure out exactly how cells buffer this iron toxicity, we may be able to discover new synthetic lethalities that now use this dependency of iron for cancer growth. And so when we started looking into ferroptosis, some of the major players, as I mentioned, are GPX4 and SLC7A11. And what we found was that indeed in tumor tissues, SLC7A11 and GPX4 are upregulated in colorectal cancer patients compared to adjacent normals. What we can do, again, as I said, is we have an enteroid bank. We can take the same tumor tissues and make enteroids from them and ask, is this a property of the epithelium? And again, what you can see in our enteroids taken from normal or tumor samples, we can again indeed see an increase in SLC7A11 and GPX4. And so it's suggesting that maybe these are the mechanisms by which iron toxicity is buffered. Therefore, these cells can now accumulate high levels of iron and grow. And so then what we did was we knocked out GPX4 in vivo in the intestine. And here what we're doing is we're using a tamoxifen inducible Cree. And the reason is that I thought if you knocked out GPX4 in the intestine, these mice would die because there would be massive ferroptosis that happens spontaneously in the intestine. And the reason for that thinking was that you can throw on inhibitors or you can knock out GPX4 in a large number of cell lines, and indeed, you see spontaneous ferroptosis. To our surprise, if you knock out GPX4 in intestinal epithelial cells, these mice are completely normal. The intestines look normal. You can stress them with almost any acute stressor, and we don't see any increase in cell death or histological difference when we knock out GPX4 in the intestine. And what I'm not showing you, just because the GPX4 data is so convincing, is that we did the same thing with SLC7A11. We have the flox mice, we made the knockouts, and the intestines are perfectly normal. If we put them through a colon cancer model, the AOM DSS, we don't see any differences in tumor formation, suggesting that GPX4 is not the major mechanism by which these cells buffer iron toxicity. I was giving this talk, it must have been like a year and a half ago, and someone said, well, your model, this AOM DSS model, it's a very benign lesions. And one of the things that we know with ferroptosis, especially some of these reagents like Avastin, they're much more susceptible in more established tumors that have a KRAS mutation. And so then we decided to cross our GPX4 flox into some more advanced models. And the way we make more advanced models is that either we knock out APC, which we call the single mutant, or we knock out APC and P53, which we call our double mutants, or we knock out APC, P53, and KRAS, which we call our triple mutants. And what I'm not going to show you is that the increase in mutational burden or knockout leads to more invasive lesions, and so much more aggressive tumors. And so if we did these and we crossed in our GPX4, we don't see any change in the survival of these mice. Again, suggesting that we don't think that GPX4 plays a role in protecting from iron toxicity in these models. And interestingly, in our single mutants, knocking out GPX4 actually makes the mice worse, meaning that it makes the tumors much more aggressive and much more proliferative than wild type mice. It's the opposite of what we would have expected. We would have expected knocking out GPX would make them susceptible and decrease tumor genesis, but which it doesn't. So then it begs the question, what are other protective mechanisms in these cancer cells? And so I'm going to show you a couple pieces of data, and then because we're running low on time, and then I'm going to address any other questions that happen. But colon cancers, and I would say selectively colon cancers, but other cancers to a smaller degree, are fairly resistant to iron toxicity by just throwing on ferric ammonium citrate. And so here's some IC50s. This is done by growth. Here's looking at by colony forming, but we're in the millimolar levels of iron before we start seeing a lot of toxicity that influences growth. And so we know that actually at high levels, we're getting cell death. And so what we're doing here is using cytox green, which is a marker of cell death. And so this dye, once it gets into a cell, will fluoresce green. Some of this cell death can be explained by ferroptosis. So we're using a ferroptosis inhibitor called ferrostatin. We can also use other compounds called leproxatin. And so we decrease some of the cell death by using anti-ferroptotic agents. So some of it is obviously through ferroptosis, but we can completely rescue it by throwing on N-acetylcysteine, which we consider our general antioxidant. And so most, if not all of this, is oxidative mediated cell death. And some of it can be explained by ferroptosis, but not all. So we took this aspects into consideration when we set up this screen. So colon cells are fairly resistant. We can throw on iron and they can grow just fine. And so then what we did is a metabolic screen using CRISPR-Cas9. So basically functional genomics. And this gives us an idea where we can knock out, in a single cell fashion, we can knock out every gene in a metabolic pathway and ask, are they more susceptible, less susceptible, or the same after we throw on iron? And so we're throwing on 2 millimolar of iron where they can grow. And so if you knock out a particular gene and they die, it makes that gene, it suggests that that gene leads to more susceptibility and could be important for buffering iron toxicity. And so basically, this is what I've explained. So you knock out a gene, a protective gene, the cell should start dying more under iron stress. And if you knock out a sensitizing gene, they should we should protect them, right? And so when you do this, and we've done this now with the metabolic library, which targets about 4,000 metabolic genes, we identify the way, and I should say the way that this screen was set up is we identify genes that are potentially protective, meaning that they induce more death when they're knocked out. One of the nice things in this screen was that one of our top hits that I'm not showing was ferritin, which, as you can see, would be intuitive. You knock out ferritin, the cells can't store iron properly, and so it makes them more susceptible to death. But one of the other interesting genes that we identified was a group of genes in heme biosynthesis, suggesting that heme synthesis could have an important regulation in iron toxicity. And again, just mapping out, I find this heme synthesis pathway very interesting. Some starts in the mitochondria, moves outside, and then comes back into the mitochondria to produce heme. And all I'm going to show you is a single piece of data that indeed, if you knock out heme synthesis by using a compound called succinylacetone, you can make the cells very sensitive to iron toxicity. So here you can see, just throwing on small amounts of iron, you can almost completely kill a cell, which you wouldn't otherwise. And we can do this by not using succinylacetone, by making knockdowns to a certain degree. We can see that same sensitivity as shown here, but not in our non-targeting hairpins. So we are still trying to figure out the mechanism. And the last slide I'll end with is this slide where we're starting to figure out the mechanism. So we thought it could be one of two pathways, right? Heme synthesis is a sink for iron, meaning you ramp up heme synthesis, it brings in more iron and it loads it onto heme, which could be a safer place to store it. Or heme is utilized for signaling. Any one of those could take place. So here is where the study that we did. So again, what I'm showing you is that you knock out heme synthesis by using succinylacetone, and they're much more sensitive to iron toxicity. If you now give them back exogenous heme, you can completely protect it, which gives us an idea that we don't think it's heme synthesis. Otherwise, this wouldn't work. So we don't think that heme is the synthesis pathway is a sink for iron. We think that heme is actually signaling. And we don't exactly know the mechanisms to it. And that's what we're trying to figure out as of right now, is that how is heme, and specifically heme signaling, protecting aspects of iron? And I think what we can rule out is certain pathways that we've looked at exhaustion. And so we know that heme can activate or manipulate BOC1 signaling, which has antioxidant properties. Itself can have some antioxidant properties, but we haven't completely figured that out as of yet. And so I'm going to end here. Sorry for going a little bit over. A lot of people to thank. Most of these projects were driven by graduate student Chesta Jain, and which were launched by another student that had eventually left our lab and was pursuing a postdoc. And with that, I can take any other questions.
Great. Thank you, Yatrik. Really a great talk. Fun to think about. Of course, I'm very interested in mitochondrial iron homeostasis. So Do you think there is a protective mechanism of accumulating iron in the mitochondria?
Yeah, we think that's what we thought at one point, was that mitochondria could be a decent store for iron, but I should say, as our data points out right now, we don't think that mitochondrial respiration or mitochondria is a place that excess iron gets stored for, I guess should say safe storing. So I should say that that's very early preliminary data as of right now. So it's still possible that those could be a sink, but we don't see evidence of it as of right now.
So at least in yeast, you can actually protect yeast from iron toxicity by overexpression of mitochondrial iron importers. So I thought maybe that might be the case in yours.
Yeah, that's what we started. That was our first thought, was that could be mitochondria be a sink, based on some of those work. And we were talking with, you probably know well, Adam Hughes at that time. We ran into him at an iron meeting, and that's what he basically mentioned. He said, you should look at that. And we did a little bit, and it didn't give us much. It was mostly negative, suggesting that maybe at least in mammalian cells, it's probably not a great sink for iron for buffering the toxicity aspects.
Great. We have two other questions. Ole, do you want to go ahead and unmute and ask yours?
Sure. Hi, Ole Klomanchuk here in Nebraska.
How are
It was
you?
really interesting talk. So I'm going to start from piggybacking on the previous questions. So have you actually looked where that iron is localized subcellular wise?
Yeah, we've done a little bit. And so when you load, like, for example, in the tissues, it's hard to, we've had difficulty doing subcellular localization of iron in tissues, but if you did it in cells and you just throw on exogenous iron in cells, it seems to be spread out to a number of different places. Now we've used this kind of the labile iron dye as a way to try to see where it's going. So we use ferro orange. A lot of it, so there's a diffuse increase staining everywhere. If you used certain dyes, so like MitoTracker or LysoTracker, we can maybe see some more accumulation in the lysosome compared to other places, which was not completely surprising. There seems to be some that gets accumulated in the ER as well, but there's not just one specific place where, at least in these mammalian cells, where we can see it gather. But I would say that all of that has been done using a labile iron dye called ferro orange. And so the question is how selective is that dye, and how good is the resolution on that dye, compared to other things that are out there. So we're limited by those interpretations.
Okay, all right. Okay, my second question is, so your roxadustat thing was pretty interesting, and that rang the bell a lot. So I don't know if you're familiar with Mick Petrus's work at Columbia. So they have very much the same result, but they blame it on copper signaling. So I'm just curious to hear your thoughts about it.
Yeah. In our hands, so in that roxadustat study, which we just recently published, you can actually completely reverse that increase in cell death that I showed with a roxadustat by just getting rid of iron or lowering the iron in the media. So we think that cell death is completely dependent on iron. We started looking at the role of HIFs in bringing in copper absorption, and I would say that a lot of our data suggests that that's, at least with respects to HIFs, we don't see much with respects to intestinal copper and HIF directly. Now there could be some indirect mechanisms, but in our mouse models, the major metal that we see is almost exclusively iron.
Okay, all right, thank you.
Great. One other question from Lorena, if you want to unmute yourself.
Hi, Dr. Shah. Really interesting data. So my question is, do you have any evidence of this iron or copper accumulation to occur extracellularly, or is just inside cells? And a second question is, if it's just or mostly inside cells. Do you think it's only happening in the enterocytes, or maybe other type of cells that populate the tumors may be storing that extra metal?
It's a great question. And so we've done this both mainly in our mouse models. And so the mouse models where we've again utilized this ferro-orange labile iron dye, which now we can use by flow, and then ask in what cells are the iron accumulating. And epithelial cells, tumor epithelium seem to be the major source, but we can also see fairly high levels of iron in myeloid cells as well. Now we're not, we don't do hardcore immunology, so certainly in macrophages, in neutrophils, we can see it, but I'm not differentiating, tumor-associated macrophages to myeloid-derived suppressor cells. Just in general, we can see some accumulation in myeloid cells as well. And recently, our plan has been to, you can basically take a tumor, squeeze all that liquid out that we call tumor interstitial fluid, and that's what the tumors get bathed with. And iron is high in that, we call it TIFs. And if you just did TIFs then you're like, what are you comparing to? So we're taking the TIFs from a tumor and we're comparing to serum. And so TIFs have much higher iron compared to their serum. Yeah. And so we think there's other places that iron accumulate as well.
Okay, Ella, do you want to unmute?
Hi, Yatrik, nice to see you.
How are you?
So my question is, a while ago, I remembered Susie Torti talked about tumor behavior and how they turned on IRP2 even in iron-rich conditions, meaning tumors behaved as if they're iron deficient, but they were actually iron-rich. And that reminds me a little bit of your situation where HIF2 alpha and accumulate a lot of iron, and HIF2 alpha is still turned on, whereas in normal tissues, it's iron deficiency that would turn HIF2 alpha on. So I'm wondering if you think in tumor HIF2 alpha potentiation or stabilization or increase in function is all related to hypoxia, or do you think they also somehow detached from normal regulatory mechanisms where iron deficiency should be the one increasing HIF2 alpha and iron replenishment should decrease it, and yet they stop sensing it or stop paying attention to it and just somehow are selected genetically for turning on HIF?
Yeah, it's a great question. So we think, and this may not satisfy it may not be a satisfying answer. We think it's probably partially by both. And so what we do know is that at least the amount of oxygen that we measure is more than enough, or would be hypoxic enough to activate HIF2. The reason I say both is that we've done a little bit with respects to manipulating IRP2 and 1 and looking at what happens to HIF activation in those settings. And it still seems to be upregulated. And so this is all done by, orthotopically putting back the tumors in the intestine and seeing if HIF2 gets activated. It doesn't get activated to the same degree. That's why I think there's both are playing a role. But the other thing that we have with the collaborator is that you can actually oxygenate tumors in vivo using specific molecular probes or using hyperbaric hyperoxia types of cages. And in all of those cases, we can actually decrease, or in those scenarios, we can also decrease HIF2 in the tumors. And so we think that's probably, there's both aspects that are probably in play for regulating HIF2. The other thing that the reason I think that is that if you take an iron deficient intestine and you measure HIF2, it's nowhere near what we see when you take a tumor and you measure HIF2. So these are very, very high levels of HIF2 that we don't see under those normal regulations.
Okay, we're pretty far past our time on it. Ella, did you want to make an announcement about the East-West iron?
Yeah, I just wanted to let everybody know that we have our usual yearly East to West Iron Club, which is kind of a North American regional iron club. And this year it's in Cincinnati, November 3rd. Carolyn Philpott is the president of EWIC right now, and Brian McKenzie is a local host organizer. So hope to see some of you there, or all of you, please.
Good. Okay, thanks again, Yatrik, for the wonderful talk. It's a lot of food for thought. Need to think about it some more. If you have other questions or you need to review this, we will post this on our YouTube website so that you can go check it out there. And next month's talk will be November 14th from Kaeyang Chen, who's going to be speaking on the identification of an intracellular heme trafficking protein. And hope to see you all then. Take care. Bye-bye. Thanks again, Yatrik. Really appreciate it.
Thank
Great
you.
talk, Yatrik.
Thank you.
Hey, Diane, can I ask you a quick question about the postdoc student exchange sending to you? Just, this is silly, but you asked for 3 things. Do you want it in a single document, or do you use 3 documents?
It doesn't matter.
Okay. All right. Thanks.
Okay. All right. Great. Okay. Take care, everybody.